I welcome all of you to this class. I welcome all of you to this class. I welcome all of you to this class. And we'll continue from where we stopped yesterday. We'll continue where we stopped yesterday. Yesterday, class, I took you through the concept of genetic engineering. I took you through genetic engineering and genetic engineering techniques. Examples which are recombinant DNA technology, transgenic, recombinant DNA technology, SCNT, somatic cell nuclear transfer, and I spoke about somatic cell nuclear transfer. I spoke about let me come again. I spoke about somatic cell nuclear transfer. I spoke about cloning, and I'll be speaking today about recombinant DNA technology. These are transgenic techniques employed in genetic engineering. Am I speaking sense? Am I speaking sense? Are you hearing me? Hello, are you hearing me? Fantastic. Fantastic. So network is misbehaving there, but we'll not give in. We'll continue. Fantastic. So I'm trying to open some pictures that I want to share with you. Okay, that's good. Come. Okay. Hello? They're hearing me, thumbs up. Oh my goodness. Oh my goodness. Okay. We'll have to contend with network. We'll- I want to share some screen videos with you that can enhance your understanding what I want to do today. Okay. Okay, fantastic. Fantastic. So yesterday I spoke about genetic engineering, concept of genetic engineering. And I said genetic engineering is also regarded as gene manipulation for the benefit of man. Am I speaking sense? Am I speaking sense? Am I speaking sense? Fantastic. I further went to speak to transgenic techniques that we needed to understand in performing genetic engineering. I spoke to cloning. I spoke to SCNT, somatic transfer. But today I'll be speaking to recombinant DNA technology. Recombinant DNA as a transgenic technique. Yesterday I spoke to different types of gene transfer methods. So we're going to start from there. Yesterday, I was speaking to you about the goods of genetic engineering. And we have a whole lot of conspiracy theories about genetic engineering. And when I say conspiracy theory, they are not based on scientific proven values, but rather than emotions. Genetic engineering has a whole lot of good for mankind. But we must approach it with ethical considerations. There is extent to which we cannot think of with natural genetic process. If you go to countries like Europe, countries in Europe, in America, there are ethical guidelines that moderate extent to which science can tinker with genetic resources. So there are government policies and regulations that moderate extent to which we can tinker with genetic engineering, so that we don't end up creating monsters. For instance, I told you CRISPR is one of the laboratory techniques in genetic engineering, especially in gene editing. And gene editing can either be addition of gene or removal of gene. So gene editing, in the real sense of it, is still genetic gene manipulation. But I was telling you yesterday, traditional genetic engineering, which deal with transfer of gene at organismal level, that is very clearly. Traditional genetic engineering method deals with transfer of gene at organismal level. What modern genetic engineering deals with transfer of gene at molecular level. Am I speaking sense? Am I speaking sense? Am I speaking sense? Fantastic. Fantastic. Now, that is why outbreeding mating systems or method are the different types of gene transfer method at organismal level in traditional genetic engineering. But the challenges we may have, especially gene editing, that involves removal of an entire region of a gene. We must approach such with care, with caution, because my knowledge of gene function is that gene does not work alone. Do not work alone. Genes work together. My knowledge of gene ontology in RNA-seq technology, where gene ontology extrapolate the function of gene function, molecular function of gene, cellular component where the gene is formed, and biological process. So when we do that, when we use David data-based annotation, visualization, and integrated discovery can tell us the gamut of functions a gene performs. A gene may be known to function for a particular character, but it plays certain roles in function of other genes. That is pleiotropy. When gene has, in fact, in genetics, pleiotropy has found a gene performing one function. Now, in gene ontology, in cytoscape analysis, using network of gene, how network of gene contributes to function of a body phenotype, molecular network of gene, molecular biomolecular species, mean the network of gene affects the function of a body phenotype. Many genes function together to affect the body phenotype. Cytoscape analysis has told us that. Gene ontology has told us that. For instance, now we are doing CRISPR. We want to cut, maybe a segment of this gene has found out it contributes to cancer in the body. And when we cut away this gene, we will solve the issue of cancer. We must approach such with care, with caution, because there are other genes located within that region that play other functions. The region you are cutting away may be accommodating other gene functions, genes, which play certain role in other gene functions, which play other role in the expression of other body phenotypes. So we are saying you want to cut this segment because this segment is accommodating gene causing cancer. And when we remove it, indirectly we'll have removed other genes that is functioning for expression of other phenotype. Am I speaking sense? Am I speaking sense? Am I speaking sense? Am I speaking sense? Fantastic. So we must approach that area with caution. Before we cut a region with CRISPR, or to eliminate the effect of gene-causing disease, we must be double so that that region is not accommodating all other genes that affect several or gamut of body phenotypes. And not only that, a gene may be causing disease function in certain condition. That same gene may be contributing to the well-being of the body in other areas. That is the complexity that is generating a lot of controversy, a lot of discussion, public discussions within the scientific world. Am I speaking sense? Am I speaking sense? So these are the things that put together ethical consideration. You move to a lot of research to find out that this area I am cutting will not contribute to, will not tomorrow create another problem within the body system. We should be careful in generating species or organisms that may become monster in the environment, that may become a threat to the ecological balance, to ecosystem. This is why we have ethical consideration in genetic engineering. All of this does not eliminate that genetic engineering, generation of GMOs, is a cost to man, but it's not of cost to man, it's of blessings to man. But we must approach it with caution. We must fulfill all ethical considerations. Part of the ethical consideration is we cannot tinker with embryo. It's a no-go area. To tinker with human embryo, to perform human gene editing is a no-go area, embryo editing. But we may end up creating a monster. Am I speaking sense? Am I speaking sense? Am I speaking sense? Am I speaking sense? If I'm speaking, fantastic. I'm not hearing from Mrs. Adebayo or Adebowale. Mr. Adebowale, are you there? If you're there, thumbs up, let me see you. Fantastic. Fantastic. Now, So we go, let me make some incursion from yesterday class. When I told you gene transfer method, I told you about vector method, example of vector method, plasmid method. I spoke about biolistic or microprojectile bombardment. I spoke about micro injection. I spoke about electroporation. Today I'm going to speak about liposuction. I spoke about liposuction. And let me quickly give you an example of liposuction. Let me check liposuction. I'm going to show you. Now, let me speak to, today I want to dwell on cloning as a transgenic technique. And at the same time, recombinant DNA technology as a transgenic technique. But recombinant DNA technology is one of the subset transgenic technique in cloning. Recombinant DNA technology is the technology used in generating transgenic organisms. And you now use cloning to multiply transgenic organisms. So in cloning, which is a transgenic organisms, we have another which is transgenic technique. We have another transgenic technique in cloning, which is recombinant DNA technology. Let me go this way. I have told you what is cloning is. Laboratory-based multiplication of identical copy of a gene or an organism. When you create millions of copies of it, all of them are having this identical copy of genes of their parent. Am I speaking sense? Am I speaking sense? Am I speaking sense? Fantastic. Fantastic. Now, when you want to do cloning, the picture you are watching is about recombinant DNA technology. And I'm going to speak to this very extensively. In cloning, you will need vector. You will need transgenic. You will need host organism. And how do we do it? For instance, you want to clone insulin gene for gene therapy in hospitals. Now we need millions of copies of this insulin gene to transfer for gene therapy to treat diabetes-related conditions. What do you have to do? You extract the insulin gene. When you extract the insulin gene, you keep it in the test tube. Am I speaking sense? Am I speaking sense? Am I speaking sense? Fantastic. Then you need EcoR1. EcoR1 is restriction enzyme. You need host organism, host bacteria. For instance, E. coli. Yesterday I talked about vectors, bacterial vectors and viral vectors, and I gave examples of them. And I told about genetically engineered vectors like yak, like bats, bacteria, artificial chromosome, or use artificial chromosome. Now, when you have got all these material sets in the lab, you'll extract, and we need plasmid as a vector, a carrier, as a vector to carry the foreign DNA. So you get your plasmid, which is circular DNA in bacteria from your typical bacteria. That is the procedure for extraction and purification of plasmid. Laboratory techniques, molecular laboratory techniques. So you have extracted your insulin gene, you have extracted your plasmid DNA. You have got your EcoR1, a restriction enzyme. So you now put the EcoR1 inside the test tube containing plasmid. EcoR1 will cut the plasmid at the restriction site, and it will cut with staggered cut, with zigzag end, sticky end. It will cut the plasmid with a sticky end, mean with zigzag like this, zigzag with my hand, zigzag, 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 zigzag. Both of them, when it's cut at the restriction site. Restriction enzyme cut at the restriction site, and it will cut with sticky end, which meaning that a zigzag end, a sticky end, a zigzag end. Then we take our insulin gene and drop into a test tube containing already cut plasmid. And the insulin is coming with zigzag end too. So it will fit into the plasmid, and we use ligation, enzyme ligase, to join, is sticking together, those seals sticking together, and will create a recombinant plasmid. A recombinant plasmid will further be introduced into the host bacteria. For instance, E. coli. And these are gene transfer methods. You can use electroporation to transfer the recombinant plasmid into bacteria. The E. coli. You can use other gene transfer methods, but we're using electroporation. How do we use electroporation? Now we want to transfer our recombinant plasmid into our bacteria E. coli. You put everything inside the test tube. You put the test inside electroporator that has two terminals, a cathode and diode. It begins to create an electrical impulse. The electrical impulse that is being created by the electroporator will make the cell membrane of the bacteria to widen. The holes on the bacteria, the holes on the cell membrane will be open. And through osmotic pressure, and in biology, osmosis is movement from region of higher concentration to that of lower concentration. So the electric impulse generated within the test tube has created high concentration. So by osmotic pressure, the recombinant DNA will move into the bacteria. Immediately, it moves to the bacteria. Those bacteria that take up the recombinant plasmid become transgenic bacteria. That is how we create transgenic organisms, because the bacteria is now carrying foreign DNA. So when will recombinant DNA technology involve using EcoR1 reaction enzyme to cut plasmid with a stagger end, and the plasmid will then introduce our insulin or the gene of interest into your plasmid. This plasmid that imbibe, that successfully take up, where the plasmid that we have successful integration of the foreign DNA onto it, become recombinant plasmid, and recombinant DNA technology. Am I speaking sense? 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 Fantastic. Fantastic. Am I speaking sense? Am I speaking sense? Now, so let us speak to this graph you are seeing here. You see, let us speak to it. You have seen how recombinant DNA technology, we use it in cloning. Now we have created transgenic bacteria. Now I want to tell you, not all the bacteria will take up the recombinant plasmid. Some will not take it up. Then we have to eliminate those who took up recombinant plasmid, those who did not take up recombinant plasmid. How do we do that? It's simple. How do we do it? It's simple. We'll subject the content to penicillin medium. We will introduce penicillin medium into that test tube. The transgenic bacteria will develop resistance to penicillin medium. So all the bacteria that have taken up transgenic, that have taken up recombinant plasmid, will produce penicillin resistant gene. So they will be able to survive inside the penicillin medium. While those bacteria that fail to take up recombinant plasmid will die. By so doing, we have succeeded in creating recombinant transgenic bacteria. Now, let us assume we have created one, for instance. Now, we set it to cloning. This is how the cloning technique comes in. So, the transgenic bacteria will begin to multiply themselves. Don't forget, bacteria multiply by asexual form of reproduction. Simply put, by binary fission. So as the bacteria is multiplying by binary fission, they're indirectly multiplying our recombinant plasmid inside of them. At the end of the day, we have millions of copies of our recombinant plasmid. And invariably, we have multiplied our insulin gene. Then we pack all of them, we carry them in the gene library. For then anytime we need insulin, we just go there, pick on bacteria, get insulin extraction, DNA extraction kit for insulin. We extract our insulin from the bacteria, then we do our gene therapy, gene transfer. Isn't it interesting? Isn't it interesting? Isn't it interesting? Fantastic. Now, let me speak to this picture that I've shared. Briefly, the incorporation of recombinant DNA technology into cloning. You see here, this is our bacteria carrying the circular DNA inside of it. So we remove the recombinant plasmid, recombinant DNA from the bacterial cell. Our circular DNA is this. We put it inside a test tube. We had restriction enzyme. Restriction enzyme will call the plasmid at a restriction site. Restriction enzyme equal R1 will call the plasmid at the restriction site. And it would call with staggered hand, zigzag end, or sticky hands. Then on the other hand, we have extracted from our body cells, of our blood, from our body, biological part from our body, insulin gene. When we have extracted our insulin gene, we cut our insulin gene with staggered hand. You see it here. So, because insulin gene has been cut with staggered hand, example of our desired gene, and we have already cut our plasmid with sticky, with staggered hand, without use electroporation transgenic organisms. Any method that we use to generate transgenic organisms can also be regarded as recombinant DNA technology, which is an example of transgenic techniques. Now, when we insert through electroporation, the gene of interest, for instance, insulin, through electroporation, as I've explained earlier, will be integrated into. When we had a gene of interest, insulin, for instance, into the plasmid, we get recombinant plasmid. That is the recombinant plasmid here. And enzyme ligase will still the sticky end together, and the recombinant plasmid will be generated. This is a component of cloning involving generation of transgenic organisms. We have generated transgenic bacteria now, and transgenic bacteria will now be integrated into cell, into E. coli, through electroporation method, as I've explained earlier, and will eliminate the bacteria that does not take up recombinant plasmid by subjecting to penicillin medium. Those bacteria that absorb recombinant plasmid become transgenic bacteria, and they will express penicillin resistant gene. They will be able to survive inside penicillin. And those bacteria, those E. coli, those transgenic bacteria, those bacteria did not take up recombinant plasmid will die. Am I speaking, sir? That is the recombinant technology being employed in cloning. Am I speaking sense? Am I speaking sense? Am I speaking sense? Fantastic. I want to show you. Let me speak to somatic cell nuclear transfer. Somatic cell nuclear transfer. Somatic cell nuclear transfer. Somatic cell nuclear transfer. That's what I'll be speaking to now. Somatic cell nuclear transfer. Isn't it interesting? I'm coming. Let me share the screen. Okay. No. Okay. Okay. Now, beautiful. Beautiful. Mhm. Now, now, you can see the screen I'm sharing. If you're seeing a thumbs up, let me see you. Fantastic. Fantastic. Now, like I told you yesterday, in Roslin Institute, we have hemophilia. I spoke about hemophilia or coagulopathy. The difference between hemophilia and coagulopathy is that they are the same thing. They are because uncontrolled bleeding, inability of blood to clot. But the two conditions speak to inability of blood to clot. But hemophilia is genetic. But coagulopathy can be caused by any condition. Anything can happen that your blood will not clot. You'll be bleeding. Anything can cause it, including genetics. So, but bleeding as a result of genetics is called hemophilia. As a result of genetic disease is hemophilia, is hereditary. Now, we have conditions where there is inability of blood to clot. And what the ingredient to address this problem is in human milk, like I spoke yesterday. So a group of scientists in Roslin Institute in England, between 1996 to 1997, performed somatic cell nuclear transfer. Nuclear transfer is an example of transgenic technique where we can clone, where we clone sheep producing human milk, and that becomes dollyship. So because it will be difficult to see women that will be submitting their breasts for milking to produce vaccine or medication that will stop hemophilia. So what came then is to clone a sheep that will produce human milk. This cloning has been conducted in guinea pigs, in pig. The first step was done where guinea pig was used to clone human to produce transgenic guinea pigs. In guinea pigs were used to produce human hearts, but and it was successfully done. And the anatomy, physiology was performed, and it found the same way, like human heart. But because of the body size of the guinea pig, the human heart it generated was small. So the same procedure was replicated on pig. We have pig successfully. Pigs that we use to produce transgenic pigs that produce human heart for human heart transplant. It is certified. It is available. So when somebody has a need for human heart transplant, just go to the pig, slaughter the pig, remove the heart, and do human transplant. So we have transgenic pigs that have already succeeded in having human heart through somatic cell nuclear transfer. Am I speaking sense? So we can have other organs of human body to be produced via SCNT to clone them in animal's body and be able to use them. And how do we carry out SCNT, somatic cell nuclear transfer? You need a donor cell. You need a recipient cell. In this case, the donor cell is mammary gland, human mammary gland, that is producing. The human is the one donating human mammary gland cell, and the recipient is the sheep that will carry the transgenic egg or fetus and the donor cell. Now, so there we have, this is a picture, this is a similar picture of how dolly sheep was produced. Dolly sheep was produced. The sheep gave us egg. Egg cell was removed from the sheep. Egg cell was removed from the sheep. And that egg cell, what happened? The egg cell became enucleated. Am I speaking sense? Am I speaking sense? Am I speaking sense? I'm giving a thumbs up. Let me see. Thumbs up. Let me see if I'm speaking sense. Now, fantastic. So the egg cell is coming with its own nucleus. We remove this nucleus from the egg cell, and it becomes enucleated. So we have egg cell without nucleus. So enucleation is the removal of nucleus from the cell. So then we have got egg cell without nucleus. Now we take mammary gland cell from human body. From the mammary gland, we take mammary gland cell. In mammary gland cell, that is donor cell for mammary gland cell. So mammary gland cell was taken from human body. And after doing that, we put in a petri dish with but the growth of the cell is arrested. The growth of the cell is arrested. Because if the growth is not arrested, that may be the cell may be growing, and there'll be cell division, simply put. There'll be cell division. There'll be cell division. Because ordinarily cell will go. If cell is not in synapse, it begins to grow. There'll be cell division. So inside the petri dish, we will arrest. There'll be arrest of cell growth. Am I speaking sense? Am I speaking sense? Fantastic. So the cell has been extracted, mammary gland cell, for instance, has been extracted from mammary cell, put inside petri dish with nutrient, but with the cell growth is arrested, so that to prohibit cell division. The donor cell, immediately we take the cell from the donor cell, donor animal, human body, mammary gland cell. As we remove the cell from mammary gland cell from the human mammary gland, we provide environment, low nutrient environment, like RPMI, nutrient age, like RPMI, produce nutrient for cell, as a maintenance ration. You have something to eat to be alive. And thereafter, we'll arrest the growth so that we prohibit cell division to happen. Having done that, we have enucleated the cell from the sheep. Now, we now use electric impulse. Both enucleated cells from the sheep, the human body, are put together inside a test tube, and that will be what we call electrical impulse. There will be an introduction of electric shock impulse to cause fusion of the mammary gland cell into the enucleated cell. And you see it here, the cell that is fusion. So that electrical impulse creates environment that will cause fertilization to happen. It will create environment that will cause fertilization to happen, and that will cause embryogenesis to take place. So immediately, and I told you earlier, I said fertilization actually happens as a result of exchange of when two cells exchange nucleus. That is called syngamy. Interaction between two nucleus is called syngamy, and that is the baseline for fertilization. So this cell coming from the human mammary gland, which is human mammary gland cell, with electrical impulse, we have fused it, has been fused into the enucleated cell. Then we have generate transgenic cell. The further introduction of electric impulse will stimulate fertilization. And after fertilization happens, immediately cell division begins to happen. And we have around 16 stages of cell development, that is in developmental genetics. Cell division, immediately after fertilization happened, cell division will begin to take place. So 15 stages of cell development will create stem cells that are totipotent. And any of those cells you pick it can grow to become an adult. After totipotent, after 16 stages of cell development, the cell becomes pluripotent, where cell can give to multiple organs. And after that certain stage, the cell will have fate determination. Then the cell will say you have to go and form the shape of the eye, go and form the shape of the leg, go and form the color of the mouth, go and form the color of the skin, go and form the color of the teeth. By the time there is fate determination, the fate of the cell is determined, it can no longer form any other organ. But between 0 to 15 level of cell development, during cell division, the cells are totipotent. They are stem cells, and they can grow up to become an individual. At that stage here, this stem cell will not prepare the sheep we want to use. We will have prepared the sheep, hormonal preparation. All the hormone that is necessary for pregnancy will have been prepared. We have prepared the cellular system of the recipient sheep, the one that will carry the transgenic egg of an embryo or zygote, will have been prepared. Then those stem cells, those transgenic embryo will not be transmitted, transferred, transfused, implant, beautiful, so to speak. You're not going to implant it inside or transfer it inside the recipient sheep. And immediately you transfer, the implantation happens. Immediately implantation happens, and the embryo begins to grow inside the recipient sheep. And the recipient comes to term. And after parturition, a sheep carrying human mammary gland was produced, called Dolly the sheep. And the sheep grows, and it was producing human milk. And the both proximate and chemical composition of the milk was analyzed, and it's exactly the same with the composition of human milk. Because the gene producing it is still the same gene producing it inside the sheep. That is how somatic cell transfer is performed. You can do it in any organ. Isn't it interesting? Isn't it interesting? Isn't it interesting? That is how transgenic organism is produced. So let me briefly let me say something about recombinant DNA technology. Okay, okay. Isn't it interesting? If it's very interesting, thumbs up, let me see you. Thumbs up, let me see. If it's interesting, if it is interesting, thumbs up, let me see. Fantastic. Fantastic. I can see Mrs. Abiola. I can see Sade. Where are the other members of the class? Where are the other members of the class? I'm coming. Let me share this. No, this is not what I want to share. I'm coming. I want to speak to this and then. Okay. Can you see this slide I'm sharing? I want to go through recombinant DNA technology again. Then, okay. We're good to go. Now, take a look at this. Recombinant DNA technology. Are you seeing this, the slide I'm sharing? Fantastic. Oh, Cherish, I'm seeing you for the first time. Whether Mrs. Cherish or Mr. Cherish, I'm seeing you for the first time. I'm seeing you for the first time. You've not been attending my class. Give yourself a thumbs down. You've not been attending my class. Give yourself a thumbs down. You've not been attending my class. Is this Mr. Cherish or Mrs. Cherish? If it is Mrs. Cherish, thumbs up, let me see. If it is Mr. Cherish, thumbs down. I'm not seeing you. I'm not seeing you. Cherish, I can't pronounce that. That's the other name. It's okay. It's okay. It's okay. Now, you say, I've said, recombinant DNA is a transgenic technique. It's a transgenic technique for creating transgenic organisms. And I've explained before. And look, I have explained this before. It is a summary of recombinant DNA technology used to generate transgenic techniques, to generate transgenic organisms. Any transgenic techniques used to generate transgenic organism can be addressed as recombinant DNA technology. So majority of our recombinant DNA technology involves generation of transgenic techniques. So I've explained this. And what are the steps? I've explained steps in recombinant DNA technology. You will need host, you will need vector, you will need restriction enzyme, you will need host, for instance, E. coli. I have explained that. I've explained that. And the foreign DNA that have been introduced into the recombinant plasmid will express the characteristics of its own gene inside the transgenic organisms. Am I speaking sense? Am I speaking sense? So I can say genetic engineering is gene manipulation process, it's gene modification process. It's gene modification process. When it's gene manipulation, it's gene modification. And so whether genetic engineering is a gene modification process, is a gene manipulation process, all is to create a transgenic organism. So you can either do this either by traditional genetic engineering or modern genetic engineering. Am I speaking sense? Am I speaking sense? Fantastic. 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 So I have spoken to cutting. When you use enzyme to cut it, you cut with restriction enzyme. And restriction enzyme cut at restriction site. And joining the DNA together with enzyme ligase. Enzyme ligase join it together. It sealed the two ends, like I've explained earlier. So the vectors are the one that carry, I told the vectors are the one that carrier of the foreign DNA is the vector. We have DNA, bacterial vector and viral vector, which I've explained so very extensively yesterday. And I told example of vectors, example of bacterial vectors, plasmid. Example of viral vector, retrovirus. And we have genetically engineered vector, bacteriophage, cosmid. We have artificial chromosome, bacterial artificial chromosome and yeast artificial chromosome. Cosmid is a combination of cohesive end of phage and plasmid. Bacteriophage is a combination when virus attack bacteria, it creates a new organism called bacteriophage. So combination of bacterial and virus gives us bacteriophage. Am I speaking sense? So I'm just giving summary overview of what I've said before. So example of bacterial vector is E. coli, Bacillus subtilis, Streptomyces species. If you can use fungi, we have fungi vector too. Example of fungi vector is Saccharomyces cerevisiae, Aspergillus nidulans. Insect cells. So we have eukaryote cell. You can use if you eukaryote cell. No, we have all of us have prokaryote cell. If we want to use a vector from eukaryote cell, oocyte, the egg, oocyte, just egg that just begin to grow. Oocyte are the eggs that you can fertilize. Mammalian cell, all organisms, protoplast, insect cell. In plant, you can use insect cell or whole plant as a vector. No, what I've said, no, this what I've just said is host organism, host. I have spoken about vector, bacterial vector, and let me come again, bacterial vector and viral vector. Example of bacterial vector is plasmid. Example of viral vector retrovirus. And we have genetically engineered vector, bacteriophage, plasmid, and artificial chromosome. Bacterial artificial chromosome, yeast artificial chromosome. We now host cells, host organisms that we can use in cloning and genetic and recombinant DNA technology. E. coli. For instance, example of prokaryotic cells, E. coli, Bacillus subtilis, Streptomyces species. And the example of fungi that we can use as host cells are Saccharomyces cerevisiae, Aspergillus nidulans, Aspergillus nidulans. And we have insect cells. In animal, we have oocyte, mammalian cells, all organisms, protoplasts. In plant, we have intact cells and all plants. These are what we can use as host organisms, host cells in cloning. So now, importance of transgenic organisms. Transgenic organism is a powerful tool. Transgenic technique is a powerful tool for studying gene expression and gene development. If you want to know how a gene will behave, just extract that gene, create transgenic inside that gene inside plasmid or other vector. In output, you can now insert the recombinant plasmid into a cell or a whole plant, or into another cell, and you begin to study the expression, to gene expression and development. Transgenic animal model in biotechnology is good model for understanding human genetic disease, or human diseases. You guys to study gene expression. You have to study to have understanding of human diseases. Several proteins produced by transgenic animals are important for medical and pharmaceutical applications. I'm talking about importance of transgenic animal is just application of transgenic organism, application of transgenic organism, genetic engineering. Application of genetic engineering. Meaning that in application of genetic engineering, when it comes to gene, you can use it to study gene expression. In medicine, you can use it to study, understand human diseases. In medicine, you can use several proteins produced. You can use it to study. You can use it in medical application or pharmaceutical applications. We can study how gene respond, how protein produced by genes respond to certain pharmaceutical drugs or certain medicinal plants or medical drugs or pharmaceutical drugs. So before we virtually commercialize the drugs, we can study the drugs. So we can use transgenic technology, or we can say transgenic technology, technology in studying all of this. So we can use gene and see how a part is how gene respond to certain drug, certain drug. So before we release the drugs for public consumption, we can study, we can use transgenic technology, we can use genetic engineering, we can use transgenic animals to study how, and in fact, we use it for drug test. In fact, we can say clinical trials. We can say for clinical trials. Clinical trials. Drug test. So recombinant transgenic animals can be used to study gene expression, to study understand the human diseases, to study, we can use for gene trials to study how how our body will respond to a certain drug. These are the clinical trials of drug tests or clinical trials. Am I speaking sense? Am I speaking sense? Am I speaking sense? So the recombinant technology, that is application. Now let me say the advances or areas of application. Areas of application. Let me say application. Importance. Okay, let me say importance. Importance of transgenic animals or genetic engineering or transgenic technology. Let me put in that form. Now, when we say, then I go to that is importance. That is use. Importance. Advantages. Importance. Advantages. Advantages or importance of transgenic animals or genetic engineering or transgenic technology. So we have seen how good they are. Now, advances, application of recombinant DNA technology. Let me say application of recombinant DNA technology. Application. Where can we use it? That is application. Application of recombinant DNA technology or transgenic or recombinant DNA technology, application of genetic engineering, transgenic technology, or recombinant DNA technology. That's application. An application means use. Uses. Uses to application. Uses to application of genetic engineering, transgenic technology, or DNA, recombinant DNA technology. That is advances and their advances in biotechnology. Advances and their advances in biotechnology. And advances, you can say, advances of transgenic engineering in biotechnology. That is advances of genetic engineering. Advances of genetic engineering. Advances of genetic engineering. That is to say, area of application of genetic engineering or transgenic technology or recombinant DNA technology. Am I speaking sense? Am I speaking sense? Am I speaking sense? Fantastic. Fantastic. Now, in medicine, we have seen its advantages in medicine, especially in studying human diseases. When a disease is associated to a particular gene, meaning that the gene is may be hereditary, may have genetic origin for its manifestation, then they be hereditary. When a gene is hereditary, then when a disease is hereditary, we mean that disease has genetic origin for its manifestation. Then we can extract the gene responsibly for that disease, create recombinant plasmid of it, we can transfer it inside an animal and study the manifestation of that disease in an animal body. And when we do that, we see the gene ontology of that disease. We study the development of that disease, and the information obtained from such study will guide management of the disease, prognosis of the disease, diagnosis of the disease, and drug development for the disease. because we have studied ex situ outside the human body. We have studied how the disease is behaving because we use genetic engineering. And it can help. It helps also in gene expression. The way the disease will manifest itself in the animal tells about gene expression. I'm still talking about medicine. Clinical trials. We use animals for clinical trials of certain diseases, of certain drug tests, clinical trials. That is advances where we have seen a fantastic advantages of using genetic engineering. In food industry, fermentation are done in traditional genetic engineering, organismally. But now we can do fermentation industrially. Wine production, beer production, yogurt production can be done industrially. Now, instead of us, we use enzyme for fermentation process. And all these are fermentation process. Now, instead of us, for instance, want to ferment, you want to ferment a fresh milk collected from a dairy cattle. It is possible for us to just introduce the gene we can culture these fermentation organisms. Fermentation organism carrying the fermentation enzyme. You can culture those organisms through genetic engineering. So instead of us to be inoculating our fresh milk with already fermented yogurt, when we have cloned thousands, millions of copies of this fermentation organism, you just fetch them and put them into. Is it the wine you want to ferment? It is yogurt you want to ferment. It is your wheat you want to ferment. We don't need to be carrying already fermented one. We have millions of copies. We have cloned. We have generated millions of copies of these fermentation organisms. Transgenic technology, genetic engineering, recombinant DNA technology will do all of this for us. So I may ask you, with an example, discuss application of genetic engineering in food industry. Can speak to wine fermentation, yogurt fermentation, production of wine, production of yogurt, production of beer. This is an example of genetic engineering. And agriculture. In agriculture, we can use genetic engineering. We can use, and I've told you how in rice, in wheat, in barley, how we use biolistic or microbiota bombardment to perform gene transfer. We create another crop that can grow faster, that show a very short generation interval, or that is sweet. We can create a mango that is sweet. We can create, and in fact, we have created BT cotton. That is the BT cotton through genetic engineering to create a cotton that is resistant to bollworm. We have created a genetically engineered beans that is resistant to Calosobacus maculatus, bean weevil. So we can create drought-resistant crops. We have drought-resistant plants all over. The plant that does not care of rainfall is under the extreme drought. They survive. That is the gene responsible for that. We can take that gene, put into a plant, and study the intervention of that gene and see the possible outcomes. We can create that. And like I told you, grafting, when you cut plant into two, the stem. We have scion and stalk. The stalk is the bottom. The scion is what you are bringing. Now, that is a traditional genetic engineering. Grafting, budding. When you have, we have what called gumosis in citrus. Gumosis is a pest that destroys citrus plants. But gumosis destroy sweet orange. But it is difficult for him to penetrate the stem of lemon. Lemon is the big one. Lime is big. Lemon is small. Lime, that big one that is not sweet, but is big. The stem is very strong. Gumosis cannot penetrate. So that is, we do what we call grafting or budding. We use the base, which is the stalk, as lime rather, because that strong stem that gumosis cannot penetrate. We divide it at the middle of the stem into two. We insert the scion into it and bind it together. This is what we're taught in secondary school. That is grafting. After a while, they'll grow and become one plant. The sweet orange is producing, it has a stem of lime rather, and the gumosis can no longer penetrate the stem. I have seen where tangerine is producing citrus, sweet orange and tangerine, meaning that the base, the stalk, is citrus, and it was grafted, it was budded with sweet orange. And let me speak to budwood, budding in agriculture. We were taught this in secondary school. There is a node where branch grow out, grow out of the plant. Then you get a scion, a branch for another plant. You create from the plant that you're using as stock. You remove a branch, you see the bud there. This happens at a tender age. So you remove, you cut it, you now bring the scion, put it into that node area and bind it together. That is budding. You create a budwood. You can have a citrus plant, a plant generating citrus and orange at the same time. So that is genetic engineering, a traditional form of genetic engineering. And environment, I have spoken to this yesterday, environmental biotechnology. When you apply transgenic techniques or the recombinant DNA technology or transgenic technology in agriculture is agricultural biotechnology. In food industry, it's called food biotechnology. When you animal molecular animal biotechnology, we have human biotechnology. Then we talk of medicine and environmental biotechnology when we use organisms in the environment. I spoke to it yesterday, where we have oil spillage. We have found a class of bacteria called hydrocarbonoclastic bacteria that feed on bacteria, that feed on hydrocarbons. That is their food. So, anywhere you see hydrocarbon, crude oil impacted area, or hydrocarbon impacted area, you see hydrocarbonoclastic bacteria there, that one that feed on this hydrocarbon. But their quantities are small. So if you want to use them for remediation, what is for remediation? For cleaning of our environment. So we can culture thousands of copies. We can do clone of thousands of copies of these hydrocarbonoclastic bacteria, release them into our crude oil impacted environment. We begin to clean them. Am I speaking sense? Am I speaking sense? Am I speaking sense? Fantastic. So you can clone them. We can clone them. We have other bacteria. Now, there is a gene that confer hydrocarbon cleaning capacity on this hydrocarbonoclastic bacteria, and that gene is called catechol gene. Catechol gene confer ability on this hydrocarbonoclastic bacteria to eat, to feed on hydrocarbons. So we can use other non-hydrocarbonoclastic bacteria. We are doing industrial remediation, internal reclamation of our land from impact from the oil spillage. Now we can use other bacteria. How can we do it? Extract the catechol gene, clone the catechol gene, create recombinant plasmid of the catechol gene, put it inside bacteria of choice, then clone them. We will see ordinary bacteria that are not hydrocarbonic carrying catechol gene. If you release them into the environment that is impacted with oil, it will begin to clean because the catechol gene will express itself. When the catechol gene expresses itself in that transgenic bacteria, it will begin to feed on hydrocarbons. And that is how remediation is done. That is how reclamation of our land from oil spillage is done. These are the things that's supposed to be going on in Niger Delta region. Environmental biotechnology. Am I speaking sense? Isn't it interesting? Isn't it interesting? Fantastic. 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 So the recombinant plasmid in medicine, I've spoken to that in medicine. In medicine, I've spoken to insulin. Example I gave was insulin, where we create the recombinant plasmid of insulin, and we create the recombinant transgenic bacteria that carry insulin. I've spoken to that. And we have recombinant vaccine for COVID-19. So it has a sort of transgenic. A novel coronavirus identified, around December 2019. It rapidly spread across the world. And we now create a recombinant vaccine with the use of recombinant DNA technology. Covishield. When I was in India, I took Covishield. Covishield is the Indian version of AstraZeneca, if I'm correct. AstraZeneca company in UK has a company in India that producing Covishield. That's not the brand name. But Covishield and AstraZeneca is the same. By the same company, but just different name. Covishield is the same drug as AstraZeneca of India, because it's AstraZeneca company from UK, the one that came during that period, came to India to establish a plant to produce AstraZeneca in India, but it's branded Covishield. Sputnik V is Russian version of recombinant vaccine for COVID-19. Am I speaking sense? Am I speaking sense? So in hepatitis vaccine is produced as a recombinant use of recombinant DNA technology. I've spoke about in food industries, in agriculture. I've spoken to all of that. I've spoken to all of that. We have genetically modified cotton, BT cotton. We have genetically modified beans that has resistance to bean weevil, Calosobocus maculatus. So I've spoken to all of this. So I'm going to give you this material to digest. So having said all of this, I have taken you through cloning procedure. I have taken you through recombinant DNA technology. I have spoken to technology. I have spoken to somatic cell transfer, which is called SCNT. Let me quickly show you something. Let me show you liposuction. Let me show you liposuction. The slide I'm sharing is liposuction. Liposuction. Liposuction is gene transfer method. It's using sperm delivery, maybe artificial insemination. It can be used in sperm delivery, where you use lipid-based reagents, such as liposome, which is an artificial cholesterol, or we use phospholipids to deliver DNA nucleic material into anchor organisms or into anchor cell. We have phospholipids. We have liposome, which is that artificially created cholesterol. Now, let me explain to you. These are nuclear material, meaning gene of interest, desired gene. This is lipofaction reagent. It can be liposome, it can be phospholipids, it can be other lipofaction reagents. Then we mix them together in appropriate volume. Then, when we had nucleic acid and lipofaction reagent together, the complexes of the two created lipofaction, where the lipofaction reagent will drive lipofaction reagent will commingle with a nucleic materials. After 12 to 22 hours, both we will have a complex of nucleic acid and lipofaction reagent, and this is what we will now use to deliver it into anchor organisms or anchor cells. Am I speaking sense? 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 Fantastic. 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 I've seen Cherish has disappeared. I can't find Cherish anymore. I can't find Cherish anymore. Okay, I can see you again. Cherish, I can't pronounce the name. So, having gone this far, I've been able to take you through. I've broadened your understanding on genetic engineering, advantage of genetic engineering, ethical consideration. There are no-go areas. You don't think with human zygote. You don't think with human cell. I mean zygote. You don't edit human embryo. You don't edit human oocyte. You don't edit human zygote. It's no-go area. So there are ethical considerations. There are government policies. So another lecturer, another classes, we're going to take the government policies and ethical policies that must guide us in genetic engineering. Am I speaking sense? Am I speaking sense? Am I speaking sense? Am I speaking sense? So, but the knowledge of this, all of this we're supposed to do, you can do in this country, but our politicians, with bad leadership, cannot create an enabling environment for us to do all of these things. They're not rocket science. It's what we can do. God needed an enabling environment, a good lab, we have companies to manufacture reagents, and this can be done. We can create genetically engineered crops that will be able to grow under our environment. And we'll study the gene expression, gene ontology, how the gene we're introducing is affecting the function of another gene, because there are gene interaction, there are gene ontology. So we should be careful introducing gene that can affect the function of another gene. Remember, there is epistasis. Epistasis is when a gene at one locus suppress expression of another gene at other locus. We may introduce a gene into an organism, and that gene can knock out the function of another gene at other locus. These are the things we have to look into. It's an extensive research before we will come and say this transgenic organism we have found is fit for human consumption. And it's a lot of money is involved. Am I speaking sense? Am I speaking sense? Am I speaking sense? So thank you for your time today. Network has been fantastic. Network is better today than yesterday. I thank you for sharing your time with me. So I'm going to get across to you when we're going to have the next class. Cherish, I thank you for coming for the first time. I appreciate Mr. Sadim and Mrs. Adebowale. Mr. Adebowale has been fantastic. She has been calling me back to back. So thank you and God bless you. We'll meet in the next class. Have a beautiful day.